The first thing you're going to do is sort the pasta by type and color. You're going to do this so that you can count the number of each type of pasta, because each different pasta represents a different microbe. Record the number of each type of pasta that you have in your data table in the initial microbiome column. To simulate antibiotic treatment, during 30 seconds, you're going to remove green and plain colored pasta. The plain colored includes the bow tie and the little swirly one. You're going to do this one piece at a time from the bag, and you're going to put each on your table. This represents the antibiotic killing these microbes. You cannot remove any of the orange microbes. After the 30 seconds, we need to know how many microbes are left over. So the ones that survive are in the bag, and you separate them by type and color so that you can get an accurate count for your data table. Now it's time to record the methicillin survivors for round 1 in your data table. So that's data table 1, column 2. There's 22 plain B. farfalle, 8 E. rotini, 5 orange E. rotini, and 5 green E. rotini. You've successfully completed the first round of antibiotics. The microbes that survive get to reproduce using binary fission. That means the surviving bacteria splits in 2. For each surviving microbe, you're going to add 1 of the exact same pasta type and color to your microbiome. So for example, we had 5 orange rotini survivors. We added 5. So you now have 10 orange E. rotini. Then you mix them all up and you're going to put them back in your bag because this is your microbiome. Be sure to record those numbers in the after reproduction on your data table. So we have 44 plain B. farfalle, 16 plain E. rotini, 10 orange E. rotini, and 10 green E. rotini. And that goes in the after reproduction on data table 1. Now, we're going to do a second round of antibiotic treatment. So you're going to remove only the plain colored pasta and the green colored pieces one at a time for 30 seconds. Each piece that gets removed represents the antibiotic killing that bacteria. When the 30 seconds is up, move the killed microbes to the side and then count the ones that are left in your bag. First, you need to separate them by pasta type and color. Then, you're going to get an accurate count for each of the surviving microbes. You will be recording each survivor in data table for round 2. So we had 25 plain B. farfalle, 9 plain E. rotini, 10 orange E. rotini, and 5 green E. rotini that survived antibiotic treatment. Each surviving microbe from round 2 gets to reproduce by binary fission. Remember, that means each bacteria splits in 2. So you're going to take one of each of the same color and type for every survivor and add it to your microbiome. Make sure to mix it together, and then you'll put it back in your bag. Record the after reproduction numbers in the second column of your data table round 2. 50 plain B. farfalle, 18 plain E. rotini, 20 orange E. rotini, and 10 green E. rotini. Begin round 3. For 30 seconds, you're pulling out just the plain and green pasta, one at a time. Each one that gets pulled out represents being killed by the antibiotic. At the end of the antibiotic treatment, put the ones that got killed to the side, and then separate your surviving microbiome by pasta type and color, and you're going to count them. Methicillin survivors round 3. 36 plain B. farfalle, 10 plain E. rotini, 20 orange E. rotini, and 6 green E. rotini. Then you double those for the after reproduction. So you have 72 plain B. farfalle, 20 plain E. rotini, 40 orange E. rotini, and 12 green E. rotini. Then you'll notice that each of the after reproduction columns in data table 1, 2, and 3 have a total at the bottom. So make sure you add up all of those. So here to do this, you'd do 72 plus 20 plus 40 plus 12. That'll give you the total number that you have. Then for each pasta type, you are going to calculate the percent that it is out of the total. So you would take, figure out the percent for plain B. farfalle, you'd take 72 and divide it by the total that you calculated at the bottom, and then multiply it by 100 and put that number in the percent after reproduction. Now that you've collected all your data, it's time to answer the analysis questions below the data tables.